Let's try it again, shall we, Dr. Ron? All right. Welcome, everyone, to Harvard's Microbial Sciences Initiative Summer Symposium. And this is the first of our many facets of microbiology seminar series. And our goal with this summer series is to illustrate how different scientists, both within and outside of academia, bring their intellect to bear on understanding how microbes shape our world and how they help us do what we humans like to do, like take care of ourselves and make fancy drugs and all that other stuff. But it's also our goal to challenge and change the status quo. Through these online symposia, we hope to make people around the world from all walks of life feel welcome and included in these conversations. What we want is we aspire to having the Harvard Microbial Sciences Initiative be a forum for international dialogue and cooperation, and a place, virtual or otherwise, where aspiring microbiologists can find mentors and advocates and friends. So please know that you're welcome. And then you can reach out to us all anytime at MSI at fas.harvard.edu. Now, we're also committed to shattering a long-held view that has plagued scientists for centuries. The preconception that scientists are, and I'm quoting thousands of people here, not funny. Now, it's true, folks. I've heard this time and time and time again, and I'll admit that some of us have a talent for curing insomnia. I get it. But there are others who are downright hysterical. But what's even more admirable, or to me, is that some of the funniest scientists I know are also among the most accomplished scientists I know. So today, it's actually my great pleasure to introduce my friend and colleague, Dr. Ron Oremland. Now, Ron began his career as a microbial biogeochemist with the United States Geological Survey in 1977, and in the ensuing decades published more than 200 manuscripts, some unbelievable work, right, which you should all go and take a look at. Ron has contributed massively to our understanding of environmental microbiology. He's delivered more than 100 invited talks. He's been on NPR, the Discovery Channel, NASA podcasts, BBC Radio. I mean, the only thing left to do was this MSI symposium. Now, in 2005, Dr. Oremland was informed that owing to the unpleasant fumes at certain study sites, that he had one of the top 10 worst jobs in science. Look it up, folks. It's a fact. But fortunately, even that knowledge didn't stop him from continuing his groundbreaking work. Dr. Oremland's expertise in microbial metabolic processes and the potential to harness those processes to remove environmental contaminants has yielded numerous patents. And his love of studying microbes in extreme environments has led to collaborations with NASA that have gotten folks to think about how to search for life beyond Earth. Now, for the next 30 minutes or so, you're going to hear about Ron's amazing research into this most amazing of elements, arsenic. And then Ron will share a bit about his other life as a CIA spy and world famous pastry chef. Now, that last bit may be a bit of hyperbole, but Ron will certainly regale us with tales of his life as a sailor, at least some of them, as a gentleman, a scholar, and a singer. And without any further ado, I am so excited to introduce my friend and colleague, Dr. Ron Oremland. Ron, the microphone is yours, sir.
Thank you so much, Peter. It's really an honor and a privilege to be on stage here for your kickoff MSI symposia. My talk is on arsenic, the microbial and biogeochemistry of metabolic exploitation of a toxic element. I use my middle name, Seth, because even though I am retired, I'm emeritus, and there are certain things I have to do to get approved, and I didn't want to go through all that rigmarole. So let's just call me the BS artist, formerly known as Ronald S. Oremland. And let's move to the right-hand side of the periodic table of the elements, which is where the things I'm going to talk about are located. And to honor Tom Lehrer, his Elements song, if you haven't heard it, you should, begins with, there's antimony, arsenic, aluminum, selenium, and hydrogen and oxygen and nitrogen and rhenium. Well, these three elements are of the four that I've been working on for the last 30, 40 years, but I would want to include tellurium. So if I had the opportunity to change that just a little bit, tweak it, considering what he did, it would be there's antimony, arsenic, selenium, tellurium, and cadmium, and chromium, and calcium, and curium, and then it goes on. I don't have enough time to sing that entire song, but the way we got into this and the way I did much of my research over my career was taking advantage of serendipitous circumstances and feeling more like a ball in a pinball machine bouncing back and forth. So the way we got into arsenic was from selenium. We had discovered that there are anaerobic bacteria out there that will take the higher oxidation states of selenium, selenium-6 and selenium-4, and use them as electron acceptors for the oxidation of organic matter. And thereby selenium is reduced to elemental selenium and is removed from solution. We isolated some of those microbes and brought them back to the lab. And in the course of that, one day when I had nothing to do, I inoculated arsenic into the media instead of the selenium, and lo and behold, the bugs grew on it, which was rather shocking. Selenium is toxic, but it's also a micronutrient. Arsenic is just plain toxic. The reason why arsenic is toxic is because in the +5 oxidation state, it's an analog of phosphate, and it will inhibit oxidative phosphorylation, which is the basic mechanism by which we make energy. In the +3 oxidation state, or arsenite, arsenic combines with sulfhydryl groups and inactivates enzymes, so it's even more toxic. Arsenic-5 is less mobile than arsenic-3. Arsenic-3 doesn't bind to so many adsorbents, so arsenic-5 is hydrologically more mobile. Arsenic-3 is less so, but it is more toxic. Arsenic is about the 20th most abundant element in the Earth's crust. It's not all that terrific in terms of its abundance, but it gets localized in many places in high concentrations, and we will talk about that later. But basically, when the Earth first accreted a little over 4 billion years ago, arsenic was more abundant in the crust. And then it's a chalcophilic element, and when it combines with sulfur and iron, for example, and forms things like arsenopyrites, those are very dense minerals, and they were brought down into the mantle, and the crust was depleted in arsenic as time went by, but they are brought back up by volcanic forces. And it's for that reason you find arsenic in greater concentrations as minerals in regions of volcanism, especially those where other precious elements are brought to the surface, such as silver and gold and platinum. There's a distillation that brings deposits up to the surface, and arsenic is mobilized by hydrothermal fluids. And this is manifested if you go out looking at rocks that have quartz in them. You can see flecks of gold if you're lucky and hit the mother lode, but also those places have arsenic. Basically, the silica precipitates into quartz as the waters cool. So arsenic exists in 4 oxidation states: the +5 oxidation state and the +3 oxidation state. Arsenate and arsenite are the most evident. There's elemental arsenic, and there's arsenic arsine, which is the form that's incorporated into many organoarsenicals. I'm going to talk just a little bit about this at the end, but mostly what I'm going to talk about is our discovery that the arsenic-5 acts as an electron acceptor for anaerobes to breathe, as you and I would breathe oxygen, and it is reduced to arsenic-3. And then there are other bugs that have been discovered that oxidize arsenic-3 back to arsenic-5 and making a living that way. Arsenic also is abundant in meteorites that are rich in phosphorus. And it's not unreasonable to understand why that is. Phosphorus is right up here in the periodic table above arsenic. And so where this is very abundant, this should be relatively abundant too. So it's been known that arsenic will kill you. It's a really good homicidal agent. It was known in 17th century France as inheritance powder, because if you take arsenic-3 and add it to water, it's odorless, colorless, and tasteless. And you can give it to the old gentleman who is the head of the manor. And if you are on the line of the heirs, you move up. Or you can give it to the taskmaster in ancient Egypt. It was known as far back as then. And you can become the taskmaster. Then in the 19th century, this guy Marsh came up with the forensic test for the presence of arsenic in tissue, and the party was essentially over. But it still goes on. People are not aware of that, but you can test for arsenic poisoning. And these are what these bugs look like in culture. And for that, I'm indebted to my associate, Jody Switzer-Blum. You start out with your medium. In this case, she isolated a freshwater microbe, Sulfurospirillum barnesii, that grows on selenate. And as it grows on selenate, it precipitates elemental selenium as this beautiful reddish color. This we got from a freshwater slough in Nevada. She also isolated from the hindgut of a termite, Citrobacter strain TSA, which can grow on arsenic. And as it converts arsenic-5 to arsenic-3, it combines with the sulfide reducing agent in the media and forms this yellow arsenic sulfide precipitate, which is rather beautiful. They're very, very striking. And you can tell when you got something cooking, basically by the change in color. And as I said, arsenic will kill you if given in enough quantity as a lethal poisoning agent. And there are millions, tens of millions, if not hundreds of millions of people in the world that drink sublethal amounts of arsenic in their drinking water supplies. That's there normally. It's what we call geogenic arsenic that's in the groundwater primarily. And given time, it will kill you, but getting there is an unpleasant, miserable way of dying. It's first manifested in this disease known as arsenicosis, where arsenic tends to leave the body in places where you sweat. So the soles of your feet, the palms of your hand, and other places. And in doing so, it leaves behind this disruptive scar. It leaves in a form that's dangerous to tissues, and lesions are formed. And imagine what this can do to your lungs or your kidney or your liver. Eventually, it can lead to death and or by cancer and other things. It's not a pleasant way. And there's sociological changes. In this case, a woman from Bangladesh. She was young and beautiful when she was married, and then within a few years, she became ugly with bad breath and all these terrible manifestations of arsenicosis, and became abandoned. Well, this is from one of Jody's isolates from Mono Lake, Bacillus arsenici selenatus, that grows by oxidizing lactate to acetate and CO2. And the electrons that are generated from this reduce arsenate to arsenite proportionally, and you get growth. If you take away the lactate or if you take away the arsenate, the bugs don't grow under these circumstances. It's very, very clear. And the stoichiometry balances very nicely. And arsenic is actually not too bad as an electron acceptor. I mean, this is oxygen, which is the strongest. If we're just oxidizing hydrogen. Nitrate is a little bit less strong. Selenate's right up there with nitrate. But arsenate is not too bad. If you were to do this on a per electron basis, -3 kilojoules per mole electrons. And it's way better than sulfate. And this is relevant because in the extreme environments I will talk about in the later slides, we can encounter environments that are very, very saline, where sulfate reduction can't occur, but arsenic reduction does. And this is just a model from Silver and Fung of the two key enzymes and the genes that govern them that are relevant here. There's arsenite oxidase, which has been known for quite some time. Arsenic-3 is brought in to this reactive center. It's a molybdenum, the reactive center of that wide family of DMSO molybdenum enzymes. And the electrons are drawn out by this risky side factor and enter the electron transport chain, where they will ultimately couple with oxygen or nitrate. And you wind up with arsenic-3 going to arsenic-5. Going the other way, arsenic-5 on the outer part of the microbe is reduced to arsenic-3 by electrons that are pushed into this enzyme complex by the oxidation of organic matter, or in some cases, inorganic substrates. And until quite recently, these were thought to be unidirectional, but we were able to show that this one can be reversed. They kind of look alike, they smell alike, they taste alike. They are bicomponents with iron sulfur side groups, but they don't do the same reactions. Now, when we were just getting into a higher gear, we were invited by science, we being John Stolz, my close colleague, over all these years. We were invited by science to write some sort of a review. And I asked him to try and line up all the microbes that we knew at that time, there's many more now, that made a living by either oxidizing arsenic-3 to arsenic-5, the red squares, or by respiring arsenic-5 and making arsenic-3, the blue dots. And phylogenetically, they were dispersed all over the tree. There was no rhyme or reason for why they were there. We even had some members of the Crenoarchaeota, these extreme thermophiles that were able to make a living by respiring arsenate. Now the key was this little critter down here, strain MLHE1, that Shelley isolated from Mono Lake. It oxidizes arsenic-3 to arsenic-5 with nitrate. And this was the first microbe we were able to get a genome annotation done for. Some of the others just fell in. There's no rhyme or reason to why they're here. And they can do a number of things. Some can also respire nitrates, some can respire sulfates, such as the sulfate reducers. The only other one that is the obligate arsenate reducer is this one that Shelley Huff McCann also isolated from Mono Lake. It's an obligate arsenitroph. It doesn't do anything but respire arsenate and uses inorganic electron donors like sulfide to get there. So there's a number of ways of looking at this. One, that this got started a long time ago and has branched out over time, or probably a bit more recently, there's been lateral gene transfer, what bacteria call sex, behind closed doors, and the genes have been transferred over all these times. And then we should have more of these guys up here. If this was a trait of the last universal common ancestor, we would expect more archaea to be present. But they're not so easy to oxidize, to isolate. But out here in California, we are blessed, if you consider a blessing, with extreme environments that are located in volcanic areas that are very rich in their arsenic content. So here's a freshwater lake, Lake Crowley, on the eastern slope of the Sierras. It doesn't have much arsenic, but if you were to go to Walker Lake, the arsenic concentration increases as salinity increases, as well as pH. Big Soda Lake, one of our first sites, is a real soda lake in Fallon, Nevada. Now we're up to 20 micromolar. Mono Lake, my favorite site, just down from Yosemite National Park, has 200 micromolar arsenic. It's very saline. And the granddaddy of them all, if you take monolake and evaporate it to a salt-saturated solution, we're talking 340 grams per liter and 3 to 5 millimolar arsenic in its dense brine waters. But let's talk a little bit about monolake. Here's a satellite view. Several million years ago, the Sierra Nevadas were pushed out from the subsurface and formed these beautiful 13, 14,000 foot range. But volcanism and plate tectonics are also involved in these processes. And there are a volcanic series of cones on the south side that started emigrating from the Long Valley caldera that erupted 25 miles to the south. And that volcanism moved northwards. It extends through Mono Lake. Paoa Island was pushed out of the lake about 400 or 500 years ago. There's hot springs on its southeastern corner. Negat is a volcanic cinder. Black Point, all these volcanic features and hot springs go into the lake. This is a closed basin lake. The basin subsided about 1 million years ago. So water goes in as draining from the Sierras, but it only leaves by evaporation. Or 15, 20,000 years ago, when the climate was much different, the Mono Lake was covered 5 times the area and had 18 times the volume of water that it does today. And it drained out of the lake and flew down, and the fluids went south all the way through Death Valley to Searles Lake that we'll talk about later. And you can see the more modern manifestations of diversion of fresh water south to Los Angeles, which cut back the amount of fresh water going in, and the lake level dropped several meters since the 1940s. Well, our initial interest in Mono Lake was on methane and other weird things that go on in this unusual environment. But then we realized it had so much arsenate in it that we decided to test how fast the reduction of arsenate occurs in the water column. And to do that, we used radiotracer arsenate and added it to water samples and incubated them. And lo and behold, we found a reduction of arsenic-5 to arsenic-3 going on at rates of between 1 to 5 micromoles per liter per day. And that there was also sulfate reduction going on at a faster rate, at a comparable rate, deeper in the anoxic water column. The water chemocline and oxycline roughly about here. So this is where the maximum rate was. And that tells you one thing, but if you integrate up all these rates, I don't know, no, I don't. The amount of arsenic reduction actually contributes to the mineralization of carbon formed by primary productivity. It can burn up 14% of annual primary productivity, while sulfate reduction accounts for about 40%. So half of primary productivity going on here by microalgae that sinks into the anoxic zone. A significant portion is burned up by arsenic. So this is the first indication that arsenic is actually more than a poison. It's an important element in the biogeochemistry of carbon in this particular very weird lake. Shelley Huft isolated this key microorganism, Alkaline-Limnicola ehrlichii. Everybody out there should say this because it's a household word, at least where I grew up. Alkaline-Limnicola ehrlichii, that I named after my professor at Rensselaer, Henry Lutz Ehrlich, who passed away just a few weeks ago, I should say. And we named it to honor him, and he was the one I blamed for getting me interested in this field. Shelley isolated using its ability to oxidize arsenic-3 to arsenic-5 and using nitrate as the electron acceptor. And here's how it does this. This is just a growth curve. Arsenite goes to arsenate. Nitrate is reduced to nitrite. The electrons balance in the stoichiometry, and you get cells. If you take away the nitrate, they don't grow. If you take away the arsenic-3, they don't grow. So it's a really nice piece of work. It's a chemoautotroph. It gets its carbon under these conditions from carbon dioxide or bicarbonate in the medium. It could also grow under those conditions with hydrogen as the electron donor, but it also is a facultative bug. It can grow aerobically with acetate just fine, thank you. And it's a really easy bug to work with once you get it in culture. That's the hard part. So we sent it to John Stolz, and he got its genome annotated, sequenced and annotated fully. And I was a babe in the woods in this. And so when I spoke to him about how things are going, he said, some very big surprises here. Well, it has ribisco, it has a reverse TCA cycle, so that's how inorganic carbon can get going. It has arsenic resistance genes. Of course, that's logical, otherwise arsenic would kill it. It seems to have methane monooxygenase, but we never could really get that going. It can definitely oxidize carbon monoxide, hydrogenase. But what about arsenite oxidase? It's not there. But that, I said, John, this is impossible. This is how it makes a living. It doesn't have what do we now call AIO-AB. Not there. But it has not one, but two genes for respiratory arsenate reductase. I said, that makes no sense. It doesn't do this. It grows by arsenic oxidation, not by respiratory reduction. Well, it turns out one of these, not both, but one of them that we were able to show works in reverse in this particular organism. And instead of calling it ARR, we now call it ARX. And this was shown by Ritchie et al. In John Stolz's lab. And working with Chad Saltykov and his student at the time, Zagar, he did knockouts. And if you take out the ARX, it cannot grow by arsenic-3 being oxidized with nitrate. Well, that was a fun exercise, got me into genome type stuff. But let's go back to Mona Lake. This is a picture that Larry Miller took. This is from Hot Spring Cove. We are in Hot Spring Cove. We're looking west. This is the other part of Paoa Island. It's just uplifted bottom sediments that have been sitting there for like 400 or 500 years. And the beautiful Sierra Nevadas in the background. But if you go along the Hot Spring Cove, you see steam because this is a volcanic region and the hot springs are getting heated from a magma chamber below. And if you get closer and actually walk on the island, you can see these beautiful rivulets of red. This is almost biblical in the sense of you expect Moses to hit the Nile and it all turns red. But these are biofilms of photosynthetic bacteria that are growing at the interface where sulfide, methane, and arsenic-3 are coming out and encountering light. We were able to isolate from this a photosynthetic bacterium that I'll talk about in a couple of minutes, but this is not a pure culture. If you take some of this material, which is very easy to scrape off the rocks with a soft toothbrush and look at it under a microscope, this is what it actually looks like. Not one, not two, but dozens of different types of microbes. In the background, you'll hear Jody and Larry being amazed by this. I mean, look at all the different types you have. I urge you, if you ever want to get into microbiology, not to just look at your pure culture, if you do that, or look at the gels or the molecular stuff. Look at this stuff under the microscope. You'll be mesmerized by it. It's incredible. It's a wonderful field. Well, Shelley isolated a pure culture, but, excuse me, this is the mat itself. If you add arsenic-3 to the mat, it will consume the arsenic-3 and oxidize it to arsenic-5 in the light. If you do it in the dark, it doesn't happen. And then Shelley isolated in pure culture a microbe that grows by it, Ectothyroidospora strain PHS1. And it does not have an arsenite oxidase. It has this ARX AB gene that we call the reverse arsenate reductase. And it's very analogous to the one we had in Alkalimnicola ehrlichii. If you go back to that mat and take samples of it now and give it some arsenic-3 in the light, it's consumed. You make arsenic-5, but as soon as you turn the lights out, the arsenic-5 goes away and you make arsenic-3. If you remember, this is not a pure culture. There are other bugs there, including arsenate respirers that make a living doing this with sulfide coming out of the hot spring as their electron donor, or hydrogen. So you have a coupled system here, just as you have for the nitrogen cycle or the sulfur cycle. Here we have my wife was just waving to me out there. Hi, Fran. Coupled arsenotrophy of arsenite oxidation and arsenate reduction, here driven by light. So in short, in nature, there's a full arsenic cycle just going from arsenic-5 to arsenic-3 and back. On the reductive side, arsenic-5 is reduced to arsenic-3 at the expense of the oxidation of organic matter or of inorganic matter, such as sulfide, where they can be chemoautotrophs and form organic matter based on this coupling. On the oxidative side, arsenic-3 goes back to arsenic-5 by the chemoautotrophic action and formation of organic matter with oxygen or nitrate, possibly by manganese as well. That hasn't been shown yet, or iron-3. But you can also do this with light. And this is the big clincher, because once upon a time, a few billion years ago, the Earth was anoxic, and the arsenic that was kicking around was arsenic-3, not arsenic-5. But if all it takes is arsenic-3 to be converted to arsenic-5 with some light, you don't need to invoke the presence of these strong oxidants. You can just do it by saying, let there be light, and we'll make arsenic-5. And by doing so, we create a niche for these guys. And that's indeed our hypothesis for this occurring. And then in this realm of thinking, you really can go off the deep end, because we're going back 3-4 billion years ago, and who the heck really knows what happens. But there's ways of making the problem tractable. Here, this is from Chad Saltykov's group, and it's a very primitive rendition of the phylogeny of the DMSO reductase family of enzymes, the so-called complex iron sulfur molybdo tungsten enzymes, the schisms. And here's the nitrite reductases and polysulfide reductases. Here's arsenite oxidase, the ones that are coupled to aerobic bugs. Here's the respiratory arsenate reductases. And over here's a branch of these reverse ones, the ones that have ARX, that take arsenic-3 to arsenic-5 by homologs of these arsenate reductases. And you can see they're not related to each other. So which came first, the chicken or the egg? And we hypothesize that these came first. They invoke light to make arsenic-5 and created a niche for these guys. And thereby they formed a conjunction. These guys came along a lot later, when oxygen became present in the Earth's atmosphere. And Michael Wells, who got his PhD just recently in John Stolz's lab and is now at Oak Ridge, has further reinforced that this goes back to LUCA. The first one to jump out of this was a formate dehydrogenase, later thiosulfate reductases, but also that arsenate reductases are ancestral genes and they arose before the aerobic arsenite reductases. That made perfect sense to me. Not to everybody, but it made sense to me. So there are even more extreme places to go. This is Searles Lake in the Mojave Desert. That's Larry Miller and Tom Cope. They're not standing on ice, they're standing on a salt crust about yay thick. Underneath the salt crust is a good couple of feet of a salt-saturated brine that's very rich in arsenic. And below that are anoxic sediments. And in here, there are bugs that respire arsenic-5 going to arsenic-3, as well as those that will oxidize arsenic-3 going back to arsenic-5. That's rather incredible that they can live under these circumstances. It's a pretty miserable place, so I only sent these guys. I've never been there. True confessions. Jody actually pulled out a microbe from this Hal arsenitibacter silvermanii that makes a living by respiring arsenic at salt saturation. It's got flagella, it's motile, but it also, if you do a thin section that Terry Beveridge did, it has this filament internally. And unfortunately, I don't have the film, but basically it moves in an eel-like fashion as a fish would, but pushing water with what would be the equivalent of its body. It's really a weird bug. I would love to see people do more with this. And here are just some shots of Searles Lake. There's Chad Soltikov and his students and postdocs. This real curious area. Well, some years ago, this paper by Sforna came out where they claimed they had evidence for a full arsenic cycle in an environment that was very similar to Searles Lake going on 2.7 billion years ago, which was before the great oxidation event. And that's been reinforced recently. So they were talking roughly about here, and there's with what Michael Wells was doing, we're talking maybe the origins of arsenate respiration going back to these good old days, well before the great oxidation event. And so well before aerobic oxidation of arsenic-3 to arsenic-5, and certainly well before the GOA, that's garlic, onions, and anchovies on pizza, which probably killed off the old dinosaurs. Okay, I would be remiss if I didn't at least talk about the infamous work we published on about 10 years ago, where Felicia Wolf-Simon came up with this very bold hypothesis that in these group 15 elements, arsenic might be able to substitute for phosphorus in the critical molecule of life, that is in DNA, not in the gene itself, but in the sugar phosphate linkage that holds the gene together. That is, in the nucleotide, it might be possible to insert arsenate into here, linking the deoxyribose sugars together on the outside of the double helix. This was a very bold idea, and I liked it. So we managed to get some money for her to come to my lab and to pursue it. And in doing so, she used Mona Lake to isolate just simply in aerobic media using glucose, this strain of bacterium, strain GFAJ, hylomonid, not a particularly unusual microbe that grew, if she grew it in saline media with arsenate instead of phosphate. It grew better with phosphate, but it definitely grew with arsenate compared to the control. And it did this just by ODs or just by direct counts. And we were very excited. We never really expected such kinds of results. And she did a number of tests to reinforce that. The final was X-ray spectroscopy, which indicated that at least the DNA extracted in whole cells, looked like it would fit what would be the signal for DNA if arsenic was substituted for phosphate. So we got a lot of press. It was published in Science. And then within a couple of days, all hell broke loose for reasons I will not go into. But two other groups that we gave the bug to could not reproduce our results. They did it by other methods. They did numerous extractions and aqueous washings, which by definition would break down the arsenic 5 ribose bond. It's not stable. That links to form the nucleotide. But in any case, we went through a lot of crap, and we never retracted it because we'll let it stand. But I would never want to repeat this, which is why I really got into drinking over the last 10 years. It helped me get through it. But there are hints in the literature that say maybe we're not entirely crazy, if not entirely right. This bacterium, Agrobacterium tumefaciens, has been shown it will import arsenic-3 under phosphorus limitations into the cell and will oxidize it to arsenic-5 and will put that arsenic-5 into places where phosphorus usually goes, such as phospholipids will become arsenolipids. And it grows better under these conditions when given the arsenic-5, very much like what we had with GFAJ. So they will use arsenate as a phosphate analog, and perhaps in doing so, free up some of that phosphate so it could carry on in the DNA molecule. Now there's this particular organophosphate, organoarsenate compound that my colleagues in Austria, Ronnie Glabonjat and Kevin Fraschesconi, have been finding all over. This is the only case of an arsenic-bound ribose molecule outside of RNA that occurs naturally. So he said, you know, we have a lot of these microorganisms in Monolake, once again, getting them involved in Monolake. In this case, Mickey, our Picocystis, this is a small, very small, 1 to 3 micron size eukaryotic algae that's responsible for more than half, if not of the primary production of Monolake. Sometimes the lake actually turns this pea green color or Kelly green. And under the microscope, under low light conditions, it forms these ears that where the chlorophyll and other ancillary pigments lie, which is why we call it Mickey, and allows it to grow at extremely low light levels. And it has these organoarsenolipids in abundance in their tissues. Finally, where you have volcanoes and water, there could be life in the solar system besides Earth. We've centered on Mars as an analog using our studies of Searles Lakes and at extremes of desiccation as a possibility. Mars doesn't have water on its surface now, but it certainly has these extinct volcanoes, and it's clear that a fluvial history occurred and that once upon a time, there may have been lakes and seas on Mars that, as it lost its volatiles, became more and more saline to the point of saturation. And under those conditions, if there was arsenic present, they would have been like the last electron acceptor that would have supported microbial life on Mars. And that's all about time I have to talk. I do need to thank the USGS for allowing me to do the work and supporting it. NASA's exobiology research program for the grants they funded. The three ladies who used to work in my lab, Jody Switzer-Blum, Sean Baseman, Shelley Hoff-McCann, who is now in her foundative days at Ann Pearson's lab at Harvard, my three little maids from school, Chad Saltikoff, John Stolz, who I've worked with all these years. Here he's doing his brine fly cha-cha at Mono Lake. Tim Hollabaugh, sampling a selenium saturated brine in the San Joaquin Valley. And the three amigos, Chuck Culbertson, Larry Miller, and myself. Without all these folks, none of this work would ever have come out or been published. And I miss them all dearly. Thank you very much. Take it away.
There we go. Thank you very much, Ron. Thanks for that great show. So now it is our turn to ask you anything we want to ask you. All right, no, we have some questions. Let's see. Are you ready, Doc?
I'm ready.
All right, let's bring it on. One question is, have microbes ever been shown to grow off of arsenic in meteorites?
Well, no, nobody's actually tried that. The hard part is to actually find a meteorite that has living cells in it and that you can scratch off. Or if you found an ancient meteorite, how would you differentiate between the microbes that you cultivate that can do this versus contamination from sitting around on the surface of the earth for tens of millions of years? Mm-hmm. But that goes back to that panspermia question of pieces of Mars bouncing off to the surface of the earth and maybe getting the earth going rather than it starting in situ here. And that was controversy that goes back to the late David McKay, who claimed that there was evidence for microbes in an ancient meteorite found in Antarctica. He didn't say what they were living on, but that was enough to spark quite a lot of controversy over 20 years ago.
Yeah, indeed.
But you still have that problem of, if it didn't get started here, how did it get started on Mars? And you can't do this on and on and on infinitum. Well, it came from Jupiter. At some point, it had to get started on its own initiative somewhere. And
it's like the origins of life shell game.
It is, which is why places like Enceladus, a moon of Saturn, which is so far away from Earth, and there's really no possibility of exchange of material between there, yet there might be signs of life under the ice of Enceladus that would rule that out. But quite frankly, if signs of life are found on Mars, I'd be really happy just for that.
Yeah.
But it's a good question.
Yeah, thanks, Ron. Do we or any animals that are in environments that are high in arsenic have any arsenic utilizing bacteria in their gut?
Yep, we do.
Whoa.
And it's been shown by Stephanie Backus at Stanford, who is finishing her PhD. She spent a little time with us that we showed her how to cultivate the bugs. And then she started getting fecal samples from all over the world, including from individuals who are suffering from arsenicosis. And she can raise cultures of arsenate respiring bacteria. That actually has a lot of health implications because the colon of humans is a fermentative region. The bugs there ferment food to gain their energy, and they usually don't do much of anything else. And they couple the fermentation products, such as hydrogen, with methanogens. And those of you who can poof out methane or also will come out of your lungs. If you're at all squeamish, you can use these cavity ring down spectrometers and let you know which group you lie in.
Yep.
But There are other normal flora there that are usually fermentative, but they have the genetic capacity to respire nitrate, for example. And if any oxygen is introduced into that system through a lesion of the lining of the colon, the epithelium, white blood cells come charging in, and with them they bring oxygen. And all of a sudden, these guys have a competitive advantage. You gain much more energy by respiring oxygen or nitrate or arsenate. And they will outgrow the other fermentative bugs that are part of your flora and screw up your GI tract microbiome. In fact, one of the symptoms of arsenic poisoning is acute stomach pains.
Whoa.
And the arsenic will also go through the lesions in the epithelium and into your bloodstream and cause havoc and eventually kill you. So yes, that's a really good question.
Wow, that's amazing.
And you got billions of cells. You don't have to go out to these exotic places. You can do really good arsenic research that has a primary relation to human health in the human GI tract. And one more thing I would like to add.
Yeah.
One of the things they found is that people in the same family drinking the same water source in Bangladesh, some people come down and eating the same food. Some family members come down with arsenic poisoning and others don't.
Okay, that's wild.
It's not clear why, but one hypothesis is just the genes, while my hypothesis, it might be due to their microbial flora in their GI tract for the reasons I just articulated.
That is very cool, Ron. All right, so I have two more questions for you. One from a summer student, and then another one will be a nice segue into other topics. Summer student question: is it common for those bacterial cells in the red biofilm to be so dispersed? Or is this bacterial cell dispersion due to sample collection and not reflective of the biofilm structure?
Very good. Stick with this kid because he or she is very smart. We dispersed on purpose the biofilm so that we can prove points. But what's not been done is to go in with an intact biofilm like you would with a microbial mat with its structure and use various electrodes and microscopes to see if there is any structure in that biofilm. It's only about a millimeter thick. It's not thick at all, like a microbial mat would be in the range of centimeters. It's tiny, but it's very possible that there is some degree of structure there, analogous to a microbial mat. But it's just so easy to take a toothbrush and structure it, and you got all this stuff. It looks like a blood sample. You bring it back to shore, and you can start processing and running experiments that work, and you get published in Science, and that's good enough for me.
And then you start drinking or not. I hear
Well,
you.
it's a celebratory drink.
Exactly. Either way. So Ron, there are so many good questions. Let me ask you before I get to what I thought was the last one. I will ask you one and ask you for a quick answer. The Halarcidobacter are so cool. Is there any follow-up work being done on those?
Not at all. I welcome anybody who wants it. We can get the bug to you. Jody is still at the lab. We've got two isolates from Searles Lake. One is Halarcidobacter with a curious hula dancer mode of mobility. And then there's a sulfate reducer that also respires arsenate. I forget its name, but she also has that. I'm sure she would love for somebody to work with these bugs who
brilliant.
And she would
so folks, if you're looking for some help, to reach out to Dr. Oremland and Jody
Switzer Blum.
Yeah, Jody Switzer Blum, of course. Go ahead and reach out to us, and we can help connect you. So if you're having a hard time finding Jody or Ron, just let us know. Now, Ron, our last question, leading into our next topic, our song and dance, as they say, is what are the most memorable enrichments over the years that should have worked based on available energy, but didn't? And also, can we please hear the element song?
Oh dear. Well, the GFAJ, that was a very difficult period. We really thought it worked, and it didn't. And there have been other disappointments. God, I think I'm having a senior moment. They've been blocked out of my memory span here, but there were errors that I made in calculations that I published on. What about cultures? I can't think of it. It will come back to me probably when I'm singing the song.
That tends to be the way this works.
But I certainly have made mistakes. And the thing about mistakes is they can be very valuable. First of all, they're humbling, because now you've appeared as an idiot to the entire world, especially in the case of GFAJ1, that was front page of New York Times, all over everything. And gosh, time to hide under a rock. But we took it like the people we properly should. We gave it out, we were honest about it, and let it subside. I would never want to go through that again.
No, I hear you, Ron, but I would say that if you're going to be pushing science and going to be working on the very leading edge, it's probably bound to happen, and that's okay. And it's I applaud you for how you shared it. And
there was another instance, which is a subject of an entire different lecture on acetylene, which we stumbled on years and years ago as a fluke. Chuck Culbertson found bugs that can ferment acetylene and make acetaldehyde and ethanol and acetate and hydrogen. And they grew doing this. And we got the first paper out, and then he isolated a bug and gave it a talk at a meeting. And Bernard Schenck was in the front row copying down everything, went back home, isolated it, and wrote the paper. And then decades went by. We had this data, and we published this cockamamie story. And it held together. I mean, it went back to ancient atmospheres where there was acetylene and maybe life on Jupiter or Titan. And that was it. And then we got some money from NASA a few decades later with Mary Wojtek and started looking at it. And we published a bit more. And then lo and behold, we got the sequence for the genome, the acetylene hydratase. And my colleague, Denise Acob, and her friends at the University of Alabama, Southern Alabama, Janet Fierst and John Sutton, started querying the genomes. Now, all of a sudden, there's only like 3 bugs in culture that can make a living doing this.
Yeah.
And they started finding now thousands of hits, because usually when you're doing a microbe genome, you're only interested in this segment of this gigantic genome that can do this. And the rest you just throw into the computer. Well, the computer still has all that stuff. So if you query the computer about acetylene hydratase homologs, all of a sudden you get thousands of hits. Now, whether it does it or not, you have to test with the microbes, but all of a sudden, this is getting tractable. Now, Michael Wells, who I mentioned earlier, has done a deep dive using bioinformatics, and he sees acetylene genes going back to Luca, way back when, 4 billion years ago, in this proto microbe that from whence we all came. And this just was a few weeks ago. He hasn't published it yet. And all of a sudden, the shake, you mean this stuff I pulled out of my tuchus out of absolute desperation to get this data set that we were scooped on, that to get something for Chuck published. I mean, it might be true, and there might be life based on this, on Enceladus's, arsenic here, acetylene here. It's enough.
That's what we call the Oremland Hail Mary.
It really was.
Let it fly. Well, Ron, in the interest of
football makes you know a little when you catch it. So.
Well, so let's wrap up by hearing the element song. How's that? Or the song of your choice.
Oh, actually, I'd rather do Fight Fiercely Harvard, another
Ladies and gentlemen, the Harvard fight song.
Another Tom Lehrer song. Oh, God. Dun, 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 dun. Fight fiercely, Harvard. Fight, fight, fight. Demonstrate to them our skill. Albeit they possess the might, nonetheless we have the will. How we shall celebrate our victory. We shall invite the whole team up for tea. How jolly. Hurl that spheroid down the field and fight, fight, fight. Dun, 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 yeah, dun, 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 dun. Fight fiercely, Harvard. Fight, fight, fight. Impress them with our proudest due. Come on, chaps, do not let the crimson down. Be of stout heart and true. Come on, chaps, fight for Harvard's glorious name, 'cause it'd be peachy if we win the game. Goody. Let's not try to injure them, but fight, fight, fight. Let's not be rough now. Fight, 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 and do fight fiercely. Fight, fight, fight. That's all I have the voice for, guys.
Dr. Ron Oremland, you are amazing. Thank you so much. And that was a wonderful talk. And folks, if you have any more questions for Ron or want to get in touch with him, just send us that email, right? At ms at I think we're where are we? What is our email? Let's go ahead and ask Hannah or someone to put it there in the chat. Ron would be, I'm sure, would be delighted to hear from you. Thank you so much, Ron. I appreciate it very much. And I'd love to have you back again. So you stay happy and well and safe out there, okay?
That's my show up there.
All right. Bye, my friend. Take care.
So much. It was a pleasure. Be well, everyone. Stay safe.
Bye.
Bye.